Imagine waking up in the middle of the night to a home invader.
Right, which is already a terrifying thought.
Yeah, totally. But this intruder doesn't just, kick down the door and smash your windows and grab your TV.
Yeah.
Instead, imagine an invader that quietly sneaks in, somehow completely hypnotizes your very expensive high-tech security system, and then physically hands hard hats to your own security guards.
Oh, wow.
Right. And suddenly your guards are paving this smooth, brightly illuminated highway leading directly to your vault, happily waving the thief right in.
Which sounds like an absolutely absurd heist movie.
Exactly. But this is exactly, down to a microscopic level, what glioblastoma does to the human brain.
Yeah, it really is.
Glioblastoma, or GBM, is the deadliest brain cancer out there. And its architectural brilliance is just, it's as fascinating as it is terrifying.
It is a complete and utter subversion of the brain's natural defenses. So welcome to the deep dive, everyone.
Yes, welcome.
Today, we are looking at a really groundbreaking study recently published in the journal Nature Cancer. It maps out the precise mechanisms of how this cancer infiltrates the brain.
And they used some wild tech for this, right?
They did. The researchers used incredibly advanced mouse models alongside actual human tumor data to show how cancer literally brainwashes the brain's resident immune cells into doing its heavy lifting.
Okay, my mind is already blown by the premise here. We're talking about a cancer that doesn't just grow as a dumb lump of mutant cells.
Right. It's not just a blob.
It actually functions like a hostile city plan or building infrastructure.
Yeah.
And our mission today is to tear down exactly how that happens step by step for you listening.
Exactly.
But before we get into the how, I have just a basic logistical question. How do we even know what these microscopic cells are doing? Like, are researchers just looking through a really powerful magnifying glass and guessing?
That is a great place to start, actually, because the technology used here is really what makes the whole discovery possible. Historically, if you wanted to study a tumor, you'd take a biopsy, so a tiny chunk of tissue, and you'd basically grind it up to see what genes were active.
Like putting it in a blender.
Exactly like a blender. But that just gives you an average of everything in the mix. It's like putting an entire city block into a blender.
Oh, I see.
You know there are apples and bricks in there, but you have no idea who was eating the apple or who was laying the brick.
Right, it's just a bush.
Yeah. So in this study, they used a technique called single-cell RNA sequencing, or cRNA-seq for short.
Meaning they look at literally one cell at a time.
Exactly. Instead of the city block smoothie, SEINA-seq lets us isolate every single cell and read its specific individual genetic instruction manual at that exact moment.
Wow.
They are looking at the cell's transcriptomic state.
Transcriptomic state. Okay, let's translate that for everyone listening. That basically means looking at what blueprints the cell currently has open on its desk, right?
Spot on.
Like what proteins it is actively building right then and there.
Exactly. It tells us what the cell's current job is, not just what it could theoretically do. And by looking at thousands of individual cells this way, the researchers found something amazing.
What's that?
They found that the cancer cells and the brain's immune cells, which are called microglia, by the way, exist in 3 distinct predictable states.
3 states, yeah.
And what's crazy is that these 3 genetic states map perfectly onto 3 physical zones of a battlefield.
Okay, I love a good map. Paint me a picture of this battlefield. What is the very first zone?
The first zone is called the oncofield. Think of this as the area far ahead of the actual main tumor mass.
Like a distant scouting zone.
Exactly. And what is incredibly eerie here is that the microglia, remember, those are our brain's immune defenders, are already changing their behavior before the cancer cells even arrive.
Wait, really? How do they normally act, and what are they doing differently in this oncofield?
Well, in a healthy, quiet brain, microglia have these long, fine branching arms. They sit there stretching these arms out, just constantly scanning the brain fluid for damage or infection.
Like little radar dishes.
Yeah, exactly. But in the oncofield, even miles away from the tumor, they pull those arms in. They become stubby and they start multiplying rapidly.
Oh, wow.
It's a preemptive state of high alert.
It's like they're pulling up the drawbridges and sounding the alarms because they sense something coming.
Right. They know trouble is on the way.
But eventually, the enemy does actually arrive. So what happens then?
Then we enter the second zone, which is called the oncoinfiltrate. This is where small scouting parties of cancer, maybe just clusters of 1 to 10 tumor cells, start pushing into the healthy tissue.
Okay, so just a few bad guys making it through.
Yeah, and here, the microglia do something really heroic. They swarm these small clusters and form what the researchers call glial nets.
Glial nets. You mean literally throwing a biological net over the bad guys?
Precisely. This is your immune system's very first line of physical defense. The microglia surround the infiltrating cancer cells, and they adopt this star-like or stellate shape.
Stellate shape. Okay.
And they orient themselves randomly in all directions, essentially trying to tangle the cancer up and entrap it like a net catching a fish.
To prevent it from moving any deeper into the brain.
Exactly. They're fighting back.
Okay, so I'm tracking so far. The immune cells sense the danger, they multiply, and when the cancer shows up, they actively fight it. They try to trap it in these nets. That makes perfect sense to me.
Right. It's what you'd expect.
But then there's a third zone, right?
Yes. The oncostream. This is the ultimate invasion highway, and this is where the paper gets just wild.
Okay, brace me.
In the oncostream, both the tumor cells and our protective microglia stretch out into these elongated spindle-like shapes. The scientific term is fusiform.
Fusiform. So they look like stretched out torpedoes.
Yeah, basically. And they line up end-to-end, parallel to each other, and move together in a massive collective migration track.
Wait, hold on. I'm completely lost. The microglia were just fighting tooth and nail to trap the cancer in a net?
They were.
How on earth do they suddenly stretch out, line up, and help the cancer build a highway? Like, why would they switch sides? That makes no sense.
I know. And that is the million-dollar question of this entire study. The transformation from a protective, tangled net to a complicit, highly aligned highway is a profound shift.
It's a total betrayal.
It really is. The cancer isn't just pushing past the microglia, it is actively recruiting them into its workforce.
How do we know that for sure?
Because the researchers saw the density of a specific protein, called Pu.1, dramatically increase as the cells move from the oncofield into these oncostreams.
Pu.1, what does that do?
Pu.1 is a master regulator that drives the massive mobilization of these immune cells. So the cancer is triggering this protein to draft our own cells into its army.
Man, that is just diabolical. Okay, this is a lot of dense information to take in, so let's do a quick mental check for you listening right now.
Good idea.
We've mapped out 3 distinct zones. You have the oncofield, the early warning zone where the guards get stubby and multiply. You have the oncoinfiltrates, where the guards throw nets over the invaders.
Yep.
And finally, the oncostreams, where the cancer breaks through, brainwashes the guards, and forms a high-speed highway.
That's the perfect summary.
So for you at home, think about this. If you were a scientist trying to design a drug to save a patient's life, in which of these 3 zones would you want to stop the cancer's progress? Do you boost the nets, or do you try to block the highway?
It's the ultimate therapeutic dilemma, honestly.
Right.
But to really answer it, to understand how the net becomes a highway, we can't just look at the cells themselves. We have to look at the physical road they're driving on. We have to talk about the extracellular matrix, or the ECM.
Right, because cells don't just, float around in empty space. The brain has an actual physical texture.
Exactly.
But I've always heard the brain described as kind of mushy, like Jell-O. If it's just soft mush, how does a tumor physically move through it?
It struggles, actually. The brain's normal interstitial space, the space between cells, is very squishy. It's rich in these soft, sponge-like molecules called proteoglycans.
Proteoglycans, okay.
But it is very poor in stiff structural proteins. For normal brain function, this squishiness is great because it helps delicate neurons connect.
Right, you don't want a hard brain.
Exactly, but it severely hinders a tumor. There is a concept in biology called durotaxis.
Durotaxis. Okay, let's break that down for everyone.
Sure. Duro means hard or stiff, and taxis means movement. It basically means that migrating cells strongly prefer stiff surfaces to pull themselves along.
Oh, I see.
Imagine trying to sprint across a pit of loose foam blocks versus sprinting across a concrete sidewalk. You need resistance to get traction.
That makes total sense. So a soft brain is actually a natural barrier to a spreading tumor.
Precisely. So to move quickly, the tumor has to physically pave a concrete sidewalk right through the foam pit. It has to change the environment from soft and random, which the paper calls an isotropic state, to stiff and highly aligned, or anisotropic.
Okay, let's lock those terms in. Isotropic means looking the same in all directions, like a tangled ball of yarn, right?
Yes.
And anisotropic means having a distinct direction, like neatly combed hair.
That's a great analogy. And the study actually details the specific biological asphalt the tumor uses to pave this combed, aligned road.
What are they paving it with?
First, they lay down fibronectin.
Which is what, exactly?
It's a major structural protein. Think of it as the rebar in concrete. It gets laid down in these long, longitudinal threads.
Okay, rebar is down, then what?
Then there's a protein called tenascin C, or TNC. And what's really insidious here is that TNC is usually only expressed in the brain when tissue is injured or inflamed and needs scaffolding to heal.
Oh, wow. So it's stealing a healing mechanism.
Exactly. The tumor forces it to form structural borders along the oncostreams, and finally, they pack collagen directly inside the streams to firm it all up.
Okay, but wait. Building all this infrastructure, laying down fibronectin rebar, TNC scaffolding, and collagen that must take an immense amount of energy and labor. Where does the tumor get the workforce to build all this?
It doesn't use its own cells, it uses TAMs.
TAMs?
Tumor-associated microglia and macrophages.
Oh, the brainwashed immune cells we were just talking about.
Oh, yes. And the paper notes something absolutely staggering here. These recruited TAMs make up to 50% of the entire tumor's mass.
Are you kidding me? Half of the physical mass of a lethal brain tumor isn't even cancer. It's our own immune cells.
Yes, half. And they proved it by looking for high levels of a marker called IBA1, which acts like a glowing name tag that says, hey, I am a macrophage.
That is insane.
If we connect this to the bigger picture, it completely changes how we view a tumor. It is not just a ball of mutant cells, it's a hijacked ecosystem.
Right.
As these TAMs are recruited, they're the ones actually restructuring the fibronectin, the TNC, and the collagen. They're the construction workers laying down the tracks.
It's like the cancer rolls into a new town, recruits the entire local police force, hands them construction hats, and makes them lay down a slip and slide made of collagen for the tumor cells to ride on. It's absolutely diabolical.
It really is. And this isn't just in mice, by the way.
It's in humans, too.
Yeah. When the researchers use spatial transcriptomics, which is basically that single-cell sequencing, but keeping the physical map of where every cell is located intact on actual human glioblastoma patients, they saw the exact same thing.
Unbelievable.
Human brains packed with activated TAMs in those stretched-out shapes, laying down stiff roads.
Okay, let's just pause and visualize this for a second because the scale is wild. You listening right now, imagine your own brain. A soft, protective, squishy environment designed to keep you safe.
Yeah.
Now imagine up to half of a lethal tumor inside that brain is made of your own cellular police force, completely brainwashed, furiously paving stiff structural highways for the cancer to spread. It's a terrifying thought.
It is deeply unsettling.
But, it leads me to a huge mechanical problem.
What's that?
If you have millions of these cells, the cancer cells and the corrupted cops rushing down this microscopic highway, why don't they crash into each other?
Traffic control.
Right. Have you ever seen a real highway with millions of cars and no lanes? It's a massive traffic jam. How is this cellular traffic controlled so they don't just pile up?
Which brings us to the star molecular player of this entire paper, a protein called plexin B2.
Plexin B2?
Yeah.
Okay, I'm guessing since the cancer hijacks literally everything else, plexin B2 is also something stolen from a normal, healthy brain?
You're exactly right. In a normal environment, plexin B2 is an axon guidance receptor.
Axon guidance, meaning for nerves.
Yes. When nerves in your brain are injured and need to regenerate, plexin B2 helps guide the immune cells to clear the debris and build repair tracks without causing a cellular traffic jam.
Oh, I see.
The glioblastoma entirely hijacks this natural injury repair pathway.
Okay, but how does it actually prevent a crash? Like, what is the physical mechanism here?
It's a phenomenon called contact inhibition of locomotion, or CIL.
Contact inhibition. So, like, if I'm walking down a crowded hallway and I bump into you, I don't just keep walking forward, grinding my face into your shoulder until we both fall over.
Right. I would hope not.
My brain tells me you've hit something. Stop your momentum, step to the side, and slide past. Is that CIL?
That is exactly CIL, and plexin B2 is the sensor that triggers that step to the side.
Oh, okay.
When cells are moving and they collide, plexin B2 is the signal that tells the cell, stop your forward momentum, align your shape with the cell you just hit, and slide past it.
Oh, I love that. It's exactly like the thick rubber ring around a bumper car at a carnival.
Yes.
Without the rubber ring, the metal cars just smash together, lock up, and grind to a halt. But with the rubber ring, with plexin B2, they bounce slightly, align their edges, and slide past each other smoothly so the momentum keeps going.
That is a brilliant way to picture it, the bumper car ring. So the researchers wanted to prove that plexin B2 was, in fact, the bumper car ring.
And he proves that.
They designed a really elegant experiment using a conditional knockout mouse model.
Conditional knockout. Let's make sure we explain every detail of that. What does the conditional part mean?
It means they genetically deleted the gene that makes plexin B2, but, and this is crucial, they only deleted it in the immune cells, the TAMs.
Oh, wow. So they left the cancer cells completely untouched.
Exactly. The cancer still had its plexin B2, but the police cars had their bumper rings completely stripped off.
Okay, so the cancer is still trying to be aggressive, but the TAMs have no rubber rings. What happened?
The results were staggering. Without plexin B2, the microglia lost their motility. Live cell imaging showed they basically lost the ability to align after a collision.
Because they couldn't do that CIL step-to-the-side maneuver.
Right. Without that rubber ring, they just crashed into each other and stalled out. And because the TAMs couldn't move properly, they couldn't organize the extracellular matrix.
Oh, the roads.
Yes. The fibronectin and tenascin C roads fell into complete disarray.
The infrastructure totally collapsed. And what did that do to the cancer itself?
The tumor could no longer invade in streams. The collective migration tracts completely broke down. The glioblastoma was forced to shift from infiltrative growth, where it spreads out deep into the brain like the roots of a weed, to bulk expansion.
Bulk expansion, meaning it just balls up.
Essentially, yes. It got stuck in its own traffic and just grew as one big round clump. And critically, because the cancer couldn't invade deeper into the brain tissue, this significantly increased the survival time of the mice.
So wait, by knocking out this one traffic cop protein, the cancer cells literally get stuck in traffic and can't leave the main tumor mass. We're trapping them by taking away their steering wheels. That is incredible.
It really is, and the physical structure of the tumor completely dictates how lethal it is. They tested this across several different models, and time and again, taking away plexin B2 forced the tumor into a localized mass.
Okay, let's pull this together for you listening right now, because I want to make sure the clinical implications of this hit home.
They're huge.
Think about this. The bumper car ring, plexin B2, lets the cells slide past each other to build invasive roads. Remove it, they crash, the roads collapse, and the tumor gets trapped as a single round ball.
Right.
But notice what we didn't do there. We didn't actually kill a single cancer cell. We just trapped them.
Which completely changes the surgical approach.
Exactly. Imagine you are a brain surgeon. Trying to cut out a tumor that has spread microscopic roots deep into healthy brain tissue is nearly impossible, right? Yeah. You can't see the roots, and you obviously can't cut out the whole brain.
Right, you'd cause too much damage.
But if you can give the patient a drug that traps the tumor into one solid, localized marble, you can surgically remove that marble. You change the entire game of treating the disease.
You do. It shifts glioblastoma from an untreatable diffuse web into a manageable, localized target. But, this triumph brings up the ultimate mystery of the paper.
Which is?
Well, we know plexin B2 is the traffic controller. But if you remember, back in the oncofield, that scouting zone far away from the tumor, the microglia, were already acting strange.
Right. The drawbridges were going up before the cancer even arrived.
Exactly. So how does the cancer turn on plexin B2 in the microglia from a distance before they even touch?
Yeah, that's a huge gap.
Yeah.
It's like a remote sabotage signal. Does the cancer have biological Wi-Fi? How does it hack the system from miles away?
The researchers knew a secreted signal had to be involved. So they took the liquid that the tumor cells had been growing in, essentially the bathwater of the cancer.
Gross, but they.
And they dripped it onto healthy microglia. Instantly, the healthy microglia spiked their plexin B2 levels.
Wow, so the signal is definitely in the water.
Right. And using a complex computational tool called cell chat, which basically maps how cells talk to each other, they identified the specific signal. The tumor cells secrete a ligand called midkine, or MDK.
Okay, so MDK is the radio signal sent out by the cancer.
Yeah.
But how does the MDK signal actually force the immune cell to turn on the construction program and produce plexin B2?
That requires a middleman, and this is where it gets highly molecular, so bear with me.
I'm ready.
The middleman is a microRNA, specifically microRNA 12-YOA, or miR-124.
Okay, I'm going to need to explain it like I'm 5 on microRNA, because that sounds like alphabet soup. What does a microRNA actually do?
Fair enough. To understand microRNA, you have to remember how a cell builds a protein in the first place. The DNA is the master blueprint locked in the vault, the nucleus.
Right.
To build something, the cell makes a photocopy of the blueprint. That photocopy is called messenger RNA, or mRNA.
Okay, mRNA is the photocopy.
It takes the photocopy to the factory floor to build the protein. Now, microRNAs like our miR-124, they don't build anything. They are like tiny biological scissors.
Scissors?
Yes. Their entire job is to seek out specific photocopies and chop them up before the factory can read them.
Ah, okay. So in a healthy immune cell, what is miR-124 doing with its biological scissors?
In a healthy, quiet microglia, miR-124 is highly active. It binds to the exact photocopy, the messenger RNA for plexin B2, and destroys it. Essentially, miR-124 is a heavy brake pedal. By destroying the blueprints, it ensures the cell doesn't accidentally build plexin B2 and start paving highways when there's no actual injury to repair.
So miR-124 is the internal security firewall preventing the construction program from running. And the tumor's midkine signal is the hack.
Exactly.
So what happens when midkine hits the cell?
When the microglia receive the MDK signal, it triggers a massive drop in the production of those miR-124 scissors.
The scissors disappear.
Right. By dropping the microRNA levels, the tumor essentially cuts the brakes. Without the scissors there to destroy the photocopies, the plexin B2 blueprints flood the factory.
And the protein gets built.
The protein levels naturally spike, and the microglia are forced into becoming pro-invasion construction workers.
I love how elegant this is. Let's build out the espionage analogy together, because this is so cinematic.
Let's do it.
The tumor is the evil mastermind. It can't breach the vault directly, so it sends out a long-distance radio frequency that's the midkine.
Right, and that radio frequency hits the security guard's receiver. It hacks their operating system and completely uninstalls their antivirus software, the miR-124.
And with the antivirus software gone, the malware just auto-runs. The plexin B2 program executes, and the guards start building the highway for the bad guys. It is terrifyingly efficient.
It really is. And the evidence for this is incredibly solid. When the researchers genetically altered the tumor cells so they couldn't produce midkine anymore, the whole hack failed.
So no radio signal.
Right. The tumor couldn't send the signal, the microglia's security software stayed intact, and they completely failed to upregulate plexin B2.
Wow. It highlights such a massive vulnerability in the tumor's whole strategy. Which leads to my final question for you listening to this deep dive.
Yeah, this is the big one.
We now know the exact sabotage pathway. The tumor secretes MDK, which drops the miR-124 scissors in your immune cells, which allows plexin B2 to spike, resulting in a highway. So if you listening were running a biotech startup tomorrow, where would you place your bets?
Good question.
Would you try to invent a drug that acts as a signal jammer, blocking the midkine before it hits the cell? Or would you try to artificially inject synthetic miR-124 back into the brain to act as an unhackable firewall? Both seem like incredibly viable avenues for future medicine.
They absolutely are, and there are actually labs looking into similar strategies right now. Synthesizing this whole journey, this Nature Cancer paper, fundamentally forces us to change how we view glioblastoma and really cancer as a whole.
Yeah, it's a total paradigm shift.
Cancer isn't just a rogue cell blindly dividing in a vacuum until it kills you. It is an active, brilliant architect. It orchestrates a vast ecosystem.
Right.
We've mapped its invasion from the distant oncofield, where it broadcasts that midkine signal, to the infiltrates, where it physically pushes through your initial immune nets.
The glial nets.
Exactly. And finally to the oncostreams, where plexin B2 drives the construction of a permanent, stiff, extracellular highway.
So bringing it all back home, what does this mean for the future of fighting this disease?
It means we have an entirely new theater of war. Traditionally, oncology has been obsessed with finding poisons that only kill the tumor cells.
Right. Chemo, radiation.
But that is incredibly hard in the brain because of the blood-brain barrier and because these tumor cells are just so resilient.
No.
But this research suggests a totally different approach.
Don't attack the cell, dismantle its infrastructure.
Exactly. If we can block plexin B2 or jam the midkine signal, we freeze the tumor in place. We shift it from a diffuse, untreatable web into a localized target that surgeons and radiologists can actually handle.
It's about fighting the environment, fighting the city planner, not just the individual invader. And, that brings me to a lingering, somewhat philosophical thought I want to leave you with today.
Let's hear it.
Throughout this deep dive, we've seen how glioblastoma expands. It uses tenascin C scaffolding, it uses plexin B2 for traffic control. But earlier, we established that these are the exact same tools and pathways your healthy brain uses to heal from nerve injuries.
They are. The cancer didn't invent them, it just hijacked them.
Right. So it forces us to ask a profound question. Where is the biological line between tissue repair and cancer progression? If a tumor is using all the blueprints of a healing wound, are these aggressive cancers ultimately just wounds that never stop trying to heal themselves, but operating with malicious, hijacked instructions?
Wow. That is a staggering way to look at it. Biology is rarely black and white, good guys and bad guys. It's often just the misapplication of normal, vital tools that causes the ultimate devastation.
A dark but strangely beautiful thought to end on. Thank you for joining us on this deep dive into the hidden architectural world of glioblastoma. Keep questioning the world around you, keep looking for the hidden mechanisms behind the madness, and we will catch you on the next deep dive.